Hi guys, tonight we're going to be learning about scientific data and I'm going to tell you a little bit about the experiment you're going to do tomorrow and how we're going to apply that to scientific data. So first off, a little bit of a background. Tomorrow you're going to be tasting a chemical called PTC or phenylthiocarbamide. It's got a bitter taste for those people who can taste it, but not everybody can taste it. So the first thing you're going to do is to taste this chemical and see whether or not you can taste it. The history behind this chemical, it was discovered in 1931 by a chemist named Arthur Fox. So he was working with the chemical PTC and he released this cloud into the air. And this nearby scientist complained that there was a bitter taste, and Dr. Fox said he didn't taste anything. So him and the colleague began to research it a little bit more. So there actually has been a gene that's been discovered that regulates whether or not a person can taste PTC. But this gene can't actually explain this tasting phenomenon. It's also that there's a possible small environmental component to tasting PTC. So people who drink coffee and tea or smoke actually might not be able to taste PTC because those chemicals, the smoking or the coffee and tea, might have caused their taste buds to die off a little bit. So whenever you taste something on your tongue, what's happening is a chemical that's in the food or the thing that you're tasting hits the taste buds, which are found in those little bumps on your tongue. The bumps themselves actually are not taste buds. Those are called papillae. The taste buds are located on the papillae, and you can see on your tongue you have different locations of those papillae. In the front, that's where we're going to be tasting the PTC tomorrow. But what happens is once the chemical binds to a taste bud, there's a nerve cell that transmits that signal to your brain, and the brain either tells you, hey, this really tastes bitter, or this doesn't taste bitter at all when you're talking about PTC. So I want you guys to think about how you think the number of fungiform papillae or papillae will vary with or change or differ with your ability to taste PTC. So if you can taste PTC, do you think you'll have more or less of these bumps, these papillae on your tongue? And if you can't taste PTC, do you think you'll have more or less of the papillae on your tongue? So tomorrow you'll be doing an experiment where you actually will be counting the number of fungiform papillae on your partner's tongue and recording that. So what we're going to have are blue lollipops. You'll be able to lick those lollipops, and the blue dye will stain all of your tongue except for those papillae, those little bumps that hold the taste buds. Then you're going to place a paper hole reinforcer near the tip of your tongue, so the front, like 1/3 of your tongue. And finally, you'll have your partner use the magnifying glass to count the number of papillae inside the hole of the paper hole reinforcer. You'll make sure you record that data down, because what we're going to do is gather all of that data and try to come up with a support for the hypothesis of whether or not you think those number of papillae are going to affect whether or not someone can taste the PTC or not taste the PTC. So tomorrow we're doing this really cool experiment with the PTC and counting the number of papillae on your tongue. But the real reason why we're doing this is so that we can start to look at and analyze scientific data. So when you analyze results, we're going to be putting this data into tables and graphs and begin to look for trends or some sort of variation between the groups. As well, when you have scientific data, you always want to make sure that you have a replicate or a way that you're repeating the experiment. We do that so in case there's any sort of outliers or things that are out of the ordinary, we're going to be able to account for that because we're going to have lots and lots of repeats, and those repeats are going to average out anything that's out of the ordinary. And then finally, we're going to draw a conclusion from that data. So does the data that we gathered in the experiment support the hypothesis that we made?